Does anybody remember the cotton-flavored grape craze that happened a couple years ago? A bunch of people were really confused of how this even occurred, how this science even came to be. However, that is one of the things I want to talk about today. I want to explain how it is possible to go from a plant, for example, in Rhabdotsis, seen in this picture, to a DNA precipitate. The process of going from a plant to DNA extracted precipitate can be separated into three major categories: physical collection integration, 1, purification, 2, and isolation, which is the third and final step. So let's go ahead and look at what the physical collection integration part of this process looks like. Sorry. Technical difficulties. Okay. First, you would only need to collect 2 to 3 leaves. It's really easy when you're in the lab to think, I want to make sure I get enough DNA, so let me over collect these leaves. However, that is not the way to go. We use a screwdriver, but instead of a metal tip, we have a plastic tip. And for each tube that you get, it takes about 5 to 10 minutes to really degrade the leaves and break it up and get this green mixture. As explained in the 1995 journal from the Plant Molecular Biology Reporter, Microprep Protocol for Extraction of DNA from Tomatoes and Other Herbaceous Plants, written by Teresa Fulton and Stephen Tolasky, they explained that the goal of this first step is to get the green mixture, because then you know that some of the DNA from the plant has been able to be released from the plant structure. So that ends the first step, the physical collection integration. However, the green mixture is not only DNA, it has a bunch of other stuff in it. So the next step would be purification. Purification first requires a buffer made of 3 different things: nucleic lysis, Sarkozy, and microprep. You would add the buffer and then put the centrifuge tube in a 65-degree water bath. You could leave it there from 30 to 2 hours. Then you would take it out and add rubbing alcohol and chloroform and centrifuge it. And this is what a centrifuge machine looks like. It spins around the tubes really, really fast so you can separate the 2 layers. After this centrifuge is used, it separates them into a clear layer, which is the aqueous layer. That is where the DNA is, and the bottom layer, which is the organic layer. And that's all the extra stuff that we don't want in this process. As explained in the 1980 journal from the Nucleic Acid Research Journal, titled Rapid Isolation of High Molecular Weight Plant DNA, written by M.J. Murray and W.F. Thompson, they explain that the aqueous layer is where the DNA is located at. So you will want to go ahead and take it out very slowly, but make sure not to puncture the organic layer. So now we will have only the clear part in a new tube, and that ends the purification step. However, even though we just purified it, we are not sure that it's only DNA in this liquid. So the next step is to make sure that you're isolating just the DNA. This is when you would add isopropanol and centrifuge it in the machine that we just saw and dump out the liquid, and you would go ahead and see that white precipitate that was on the first slide. You repeat this process with isopropanol and ethanol. Centrifuge it and dump it out in the sink once more. And lastly, you would go ahead and put it in a speed vacuum, which looks like this. While it does turn the centrifuge tubes around in the same motion as the last machine did, the speed vac is different because it actually dries out the tube. So you want to leave the tubes open so the little liquid that you have left can be dried out. And the only thing that's left is that white DNA precipitate. As explained by the 2014 journal published by Adam Henley and Robert J. Hansey, A Simple Method for Extraction Next Generation Sequencing Quality Genomic DNA for Plant Species, you would then use an RNase mixture to make sure that the only thing you have in that precipitate is DNA and no other extra molecules. So let's go ahead and take a step back. First, you would have the physical collection and degradation. Then you would have the purification. And lastly, you would have the isolation step. And this is how it's possible to go from a plant to DNA precipitate. While it's easy to think that knowing basic scientific procedures don't really relate to you, that's not true. Let's go ahead and go back to the cotton-flavored grape craze. While a bunch of people thought it was cool, a lot of other people were really put off by the idea that a lot of sciencey stuff was happening to fruits and vegetables. However, as seen by the sources I cited, the processes that go behind plant science have been around for a really long time. I wanted to explain this procedure because I think it's important to make scientific knowledge like this common knowledge. It's important for you to understand how this happens and know that this process has been heavily researched and replicated many times in many different labs, and know that plant science is headed in a great and secure direction. As college students, we are all here to learn. So I urge you to learn more about science and to open your minds to what plant science has been doing.